afternoon, everyone. Welcome to the second day of BioKorea 2020 Live Talk. Thanks for tuning in our channel. I'm so happy to eat with you here today. My name is Kyungmi. Well, are you looking for experts in bio industry, or are you seeking to improve your business? Then this is the right place where you can join global companies through Business Forum, that is online meeting matching system of BioKorea 2020. So please visit our website for more details. As the biggest bio convention in Asia, BioKorea 2020 is delivered online convention from e-conference, virtual exhibition to business forum, including online meeting, company presentation, and especially this pioneering move, live talk, which is attended 28 companies provide opportunities to find innovative technologies, products through real-time online streaming. And this live talk is being conducted as a real-time streaming service. For English speaker, please search BioKorea at YouTube here. And for simultaneous interpretation service into Korean will be provided on channel KBIC, 보건산업혁신창업센터 for Korean people. And our channel has real-time live chat, and I look forward to your comments and any questions and feedback. An audience participating in our live talk will go in the draw for gifts at the end of the day. So please be with us until the end. So today we've got a total of 8 companies lined up today. And I am so pleased to introduce the first company of the second day of live talk. I'd like to introduce our first company, APAC Health Company, APAC Health. And this company is a private Australian-based global business that focuses on sales growth and maximizing profit. So we've invited Dr. Chris Soteropoulos. Are you there? Hi.
Yes, I am. Good afternoon, everyone. Very well.
Good afternoon. Thank you very much for joining us. So why don't you say hello to our viewers?
Hello, viewers in Korea and elsewhere.
Thank you very much. I'm getting more curious about your company. So from now on, please introduce your company.
Thank you very much. We've entitled our company motto, Great People Make Healthy Companies, because over the many years that we have worked in this space, we have observed that really it comes down to people with good values, good integrity, as well as great products and passion to help the community at large. Next slide, please. APAC Health is a company of diversified services. We provide commercialization, clinical development expertise, funding, international distribution, strategic management, marketing, recruitment and training, and also business development and legal services. The reason why we do that is that we have members of our team who have worked in big pharma, biotech, legal, accounting, regulatory, distribution, sales and marketing. So we can bring together many different elements from not only Australia, but from around the world. So even though we are located in Australia, many of our colleagues and partners are scattered around the world. Next. You can see on the diagram here the APAC Health footprint. This footprint, what does it mean? It means we have networks close to government, financial funding communities, distributors, advisors, people who are key opinion leaders, access to hospital leaders as well, so that we can identify amongst our footprint various expertise that we can draw around for ourselves, but also for our clients. Next slide, please. Our focus is on niche, new and innovative products and services. We're very excited about products that provide, have an international competitive attribute. We provide consulting services for products and services, whether they be molecules, monoclonal antibodies, medical devices, or even virtual reality. Our faster and increased sales focus is very attractive to clients. What does that mean? That means we are able to conduct a quick medical, commercial, and regulatory validation. How do we do that? Well, we call the people from different parts of the world and we ask them, can you please find out from your local hospitals, what are the prescribing protocols? What are the costs for these treatments or options? How long would it take to register? What else is in the marketplace? And we can get those answers within a couple of weeks from over 20 countries. That's very important for our clients because they need quick information and they want to know how to position their product to increase sales. Our team has many years' experience. As I said earlier, they understand the drivers of revenue. Now, the next slide is one of our magnificent products and services that we are taking to the world at the moment. We've partnered with a Basel Swiss firm called Lares Life Sciences. They provide expertise to Big Pharma on how to structure their clinical development plans, and they work closely with them. APAC and Lares have worked together to create a suite of products for both biotech companies, but also the investors. Now, why is that relevant? Next slide, please. The two broad product groups are a reference CDP, or clinical development plan, and the biotech clinical development plan. What is a reference CDP? Let's say a company wants to develop a product in Alzheimer's. The reference CDP will report on all successful products in this space, all approved products. What trials have they done? How have they designed their trials? What have they thought about in the design of the trial when you're looking at recruitment, when you're looking at cost, when you're looking at location, when you're also looking at what the regulator has to say? And as we know, regulators from China, Japan, Europe, Australia, US have slight differential issues. So with that document, you then have a very powerful tool to adapt and reflect on your internal program. And this is the second suite called the Biotech Clinical Development Plan, where Lares will work with the company, the biotech, in order to tailor, come up with considerations, identify what are the key emerging practices in healthcare or statements made by regulators which need to be tweaked to the previous protocols. Next page, please. So this is just a little bit more information on those two suites. So we said the reference CDP, overview of approved products, current industry status, clinical development plan for each product. Now, the biotech CDP is fascinating because you're also looking at patient population optimization and maneuvering and updating as you achieve the different milestones. So you are then able to refine your product vision through real-world experience. What is happening in the clinic? And this term TPP, target product profile, what you want the product to look like and achieve, needs to also be aligned with your clinical development plan. And that requires facilitation of communication with commercial organizations. and monitoring the product valuation and optimization as well. Next slide, please. This is an interesting slide. So we ask ourselves, investors, what do investors want? Well, we all know they want to reduce their risk. They want to increase the valuation, and they want a product that can be registered. So often when we speak to our investor groups around the world, we ask them, as part of your due diligence, what are you really good at? And they said, we are excellent at assessing the people, the market, the positioning of the product in the market, the scientific team involved, excellent. We've got regulatory advisors, but there's one area which we don't know very well. It's a mystery box. We open the box and there's the clinical development plan. We don't know whether the plan is going to be knocked back at FDA or other regulators because more information is needed. We don't know that. And we don't really have a handle on a lot of people who can tell us whether that's the case. Luckily, within Lares, we have developed products which help substantially to demystify for investors this area. Again, why is this important to an investor? Valuation of assets. This gives you an extra value point. Licensing. If you think your product or the company you invested in is going to receive a $200 million license deal, and then the group that comes in from Big Pharma employ their really smart, stringent people, due diligence people, they might come back and say, guess what? You've made a few mistakes in the clinical development plan, and from 200, it's only worth 50 or 80. And then everyone's hunting for the bad person. Investment. If you want to invest into a biotech, this is something you should also look at as part of your due diligence. You want to sell a stake. Again, you want to make sure whether you're not selling for an undervalued amount or not, or if you want to sell a company. So this table outlines for the biotech CDP and the reference CDP how it can assist. Now, at the execution stage, there's a biotech health check that you'll see on the slide. What's a biotech health check? That's where Lares comes in and looks at the biotech, looks at what it's either done or about to do, and it says, right, guess what? You have done really well. It has a checklist. It's like servicing your car. When you take it to a mechanic, and at the end of the service, the mechanic gives you a list with 20 different items and said, yes, that's fine, that's fine, that's fine. Oh, that's not fine. Oh, we needed to change that. This is like a ratings report, which is essential to investors in order to define what needs to be tweaked or funded in order to get a higher valuation. Next slide, please. Now, biotechs, well, of course, they also need to, at best practice, should be that they obtain a reference clinical development plan for the reasons we've outlined before. And then, should they wish to, adapt it themselves or use Lares to help them adapt it to their needs. This is a great way for biotechs that are cash-strapped to get confidence and provide confidence to investors as well. So you'll notice that there are 3 columns in this table. Status check. What is the status? Ongoing advisory, where you'd require assistance. And the big question for everyone is, yes, but is this expensive? And the way that we've priced it, it's very competitive. In fact, prior to COVID-19, I would have said it's the price of going to a conference. The flights, the accommodation, time away from office, etc. So it is very cost effective, and we think it would make a lot of sense for the biotech industry, where at the moment, and in the past more particularly, we've seen a lot of fragmentation. A lot of biotechs, experts who are maybe good at one little area but not another. Then you go into funding challenges. Funders sort of, I'm not sure. You go into licensing, you get a surprise because you never get what you want. Go to the regulators, and regulators say, oh, why have you done it that way? So this is a great way to check that. Next slide, please. So APAC, in addition to that, provides a wide range of consulting services. Let's say you have a product or service that you want to enter a market, Europe, Singapore, Australia, US. How do you really find out what the pain points are? The cost of doing it, the price, how many sales reps do you need? Who in the hospital do you need to ensure is favourable for your product? And what product are they going to not take on in order to take yours? Are there tenders? So we can look at that, but we also provide comments on innovation. So we might say, you know, what you've done there with your telemedicine is great, but can you also add another feature? Because we're being told by our friends in Poland or Belgium that that'd be nice to have. Now, that's not market information, but we know it's market information. We also introduce you to finance, different parts of the world at different stages, whether it's $300 million, $400 million, or $500,000, depending on your needs. Next slide, please. Recruitment and training. Now, this is an area which my colleague Newton Chai is leading. He's a big pharma, he's run sales teams, launched products, he knows the space very well. APAC has evolved into this area because a lot of people are coming to us from our international network saying, hey, are there any board positions available? CEO positions, sales positions, manufacturing positions, nurses for clinical trials, logistics, because you've got networks, can't you make a few phone calls and see what you can do? So that's Newton's expertise. The key there is that we spend a lot of time on culture, because as we all know, within a business unit of a biotech or a pharmaceutical, the culture may be vastly different to that of the organisation. We have to understand that, and we spend a lot of time on that in order to also select a candidate who fits in that role. Training. We're all being told that's important and more prevalent. Face-to-face, online, but at all levels of organisation as well. Thank you. Next slide. Well, that's the end of my presentation. And before I sign off, I wanted to thank the BioCareer Organising Committee and all the staff and IT staff that have worked tirelessly over the last couple of days to make this a magnificent conference. We wish, unfortunately, or fortunately, we wish we'd be there. And hopefully we'll be there in the next couple of years. So thank you very much for me.
Thank you. Yeah, as you mentioned, I hope to see you next year in Korea and the venue. Okay, thank you very much, Dr. Sotiropoulos, for your wonderful presentation. And I'm checking our YouTube channel, and Ms. Min Hee Kang has kindly asked a question. So your presentation is quite interesting. So what was the most memorable consulting in your business?
The most memorable consulting, thank you for the question, was one where a COVID-19 test was approved in Australia, and we wanted to make it available for a European country. So we spoke to that European country, and they said, yes, no problem, tests, we get it, but we also want data analytics. We want someone to evaluate the people that are testing, that are receiving the test. Where are they from? Where are they going? So we went back to the company that referred us to the test, and we said, can you please build a data analytics platform? This is memorable because it's 1 week old information. So you don't know sometimes what the customer needs unless you speak to them. Thank you.
Thank you very much for your kind answer as well. So yeah, it's time to say goodbye, Dr. Soteropoulos. Thank you once again for your time and your presentation. Hope to see you next year.
Thank you
Have a
very
good
much.
day.
Pleasure.
Thank
you.
you.
Bye-bye.
Okay. So let's move on to the next company, viewers. So again, don't be shy to leave your comments and questions. Anything is fine. Also, you can win some gifts if you participate our channel actively. So next company I'd like to introduce Accelera Canada. It is a full-service fund providing end-to-end commercialization for emerging biotechnology, life science ventures. So this time around, Mr. Michael Cloutier will give us more detail. Hello, Mr. Cloutier. Can you hear me?
Yes, hello.
Hello,
And
good to meet you here.
good morning, good afternoon, or perhaps even good evening to our folks in the audience. I want to begin by thanking the folks at Bio Korea for inviting us this evening or today, this morning here in Canada, to present our capabilities and to talk about the Canadian market in terms of the viability and the attractive nature of the market. Accelera Canada is a full-service consulting practice designed specifically to accelerate the availability of innovative products for Canadian patients, caregivers, and healthcare professionals. We have been active for over 3.5 years now and are very proud of our track record and in ensuring that a number of molecules have come to Canada quicker and in a much more efficient manner than they might have had we not been available to our clients in the US and Europe. And we're excited today to be speaking with you and hopefully to be able to create some further excitement for all of you about the Canadian market and to attract individual companies from Asia Pacific and in particular from the Korean market. So thank you for that opportunity today. If I could have my next slide, please. As I mentioned, we have well over 200 years experience amongst the 5 partners in our organization. We have experience with large pharmaceutical multinational firms, the likes of EMD Serono, AstraZeneca, Pfizer, Pharmacia, GSK, etc. Many large companies. We've worked with mid-size organizations and with a number of startups in both the pharmaceutical and biotech space. And so we bring a unique blend of experience in terms of our depth and breadth in the Canadian market, but each of us has experience in global markets, and that creates a filter for us to fully understand the needs of organizations that come to Canada because they're global organizations and they see Canada as an important market. But we realize that there are resource requirements necessary to be effective in our market. And so having that international experience helps us to understand where Canada will play and how we can most effectively and efficiently bring your innovation to the Canadian market. If I could have my next slide, please. The process that we undertake really is from 3 perspectives. We, first of all, ensure that we are providing the background necessary to make the best strategic decision on your entry into the Canadian market possible. We collaborate with you as our clients to fully inform your decision. And we do that in a way that is unique in the Canadian marketplace. We are the only consulting practice with the, as I mentioned, the depth and the breadth of experience in launching products successfully. And in terms of all elements of the value proposition necessary in order for you to make that best strategic decision. We know that there are basically three options in entering Canada. You can look at establishing a subsidiary of your organization with a full footplate and complete responsibility on your own for the P&L of the innovation that you're bringing to the organization and really starting the company up in Canada for the very first time. Or you may choose a hybrid partnership, and that is the sharing of the resources necessary to bring the product to Canada and also sharing the revenue and the profitability of that asset as it comes into the marketplace. And then third, of course, the option always exists to out-license to a partner in the marketplace and to really relinquish the rights to the product in the marketplace. Now, that third option is generally for organizations that are very strapped in terms of the resources that they have available, both in terms of time and money, and their interest in other markets that are of greater importance to them from their overarching corporate strategy. And so our goal is to fully inform your decision-making process. We don't make the decision, but rather we help to inform the decision that you will make by providing in-market, on-the-ground information necessary to have everything that you will need. If I could have my next slide, please. We fully integrate our capabilities from, three perspectives. As the slide comes forward, you'll see that we, first of all, as I mentioned, that we look to our experience. We are known as Canada's insiders. We have the information with our experience on the ground. As I said, well over 200 years in combination of all of our partners. And we gather that information both in terms of our historical perspective and experience with the latest data. And we use that insider information to really be able to provide the template necessary for a full and robust understanding of what will be necessary to be successful. We are expert builders. We have a very rich complement of partners and employees who have been there and done that, to be colloquial, in terms of the experience necessary to be effective and efficient in launches with a very high success rate. And finally, we have the capability for full end-to-end commercialization. And if you can click to the next slide. In fact, you'll need to click 2 slides forward. We're still stuck on a slide here. Yes, if you could go past this slide as well, please. In this coming slide, we have a full graphic depiction of that full complement of capability in what we call the end-to-end commercialization plan and the customized services necessary to ensure optimal success in the marketplace. If it's possible, if you could click to the next slide, I'd appreciate it. So, of course, a full and robust market analysis, which of course always includes a full understanding of what we would anticipate in terms of the label that will be achieved in the negotiations with Health Canada from a regulatory perspective. Obviously, a full and robust understanding of the market conditions relative to pricing, the market access and reimbursement market. It's a commonly misunderstood situation for a lot of outsiders from Canada that we are in a payer-only, government payer-only market, and that's not the truth. We are very much a hybrid type market. There is a great deal of third-party private insurance payment that occurs for prescription products in Canada. And so much like the United States, it's robust, but also much like Europe, there are some publicly funded elements to reimbursement. And so we have the capability in fact, probably the best track record in all of Canada for success in meeting the needs of both the private payers and the public payers. And so we have that, obviously all of the elements that are necessary related to advocacy in terms of both the healthcare professional advocacy and patient advocacy, and then all of that's necessary for stakeholder engagement, corporate setup in terms of all of the necessary components should you decide to pursue setting up a subsidiary, and then really all of the elements of both the corporate launch and the launch of the asset itself, the innovative product that will be available to healthcare professionals, patients, and to caregivers. If I could ask to have the slide forwarded again. And we'll need to actually go one slide forward again. So I want to summarize and leave some time for Q&A. The highlights really are this. We have the seasoned professionals and the market expertise, which is unparalleled, to be able to work with you as your chief consulting practitioners on the ground to help inform the best quality decision in terms of the strategic plan that will be necessary to effectively enter the market, whether it be setting up that SUD, looking for the hybrid partnership, or an out-licensing opportunity. We have the expertise both in-house and through a quality of network that's unparalleled in our industry in Canada to ensure that you will have the very best and the very brightest associated with you, and the ability to bring a very customized view of Canada, which will lead to a very customized and tailored plan for market entry into the country. We have the precision and the strength to apply those activities and that tactical plan should you choose to not out-license, whether it be in terms of your subsidiary setup or your hybrid partnership. And then finally, we will ensure that the team that's in place will be designed specifically for your needs, your product, and the marketplace as it pertains to that product. And again, in an unparalleled and very unique fashion that is only offered through the expertise that we have at Accelerate Canada. So from a very high level perspective, that is the value proposition that we bring: a complete end-to-end commercialization, strategic analysis, planning processes, and tactical implementation to ensure maximal effectiveness, maximal efficiency, and optimizing the asset for the greatest level of profitability for you in the Canadian market. And with that, I will turn things back over for Q&A.
Okay. Thank you very much for your presentation. So we will listen to Accelerate Canada. So now I'm checking the question and comments. Okay. So we have many questions. Actually, your company is quite popular among our viewers. So hi, Yuna. Hi, Minnie. Thank you for your wonderful questions. Also, we have Minhee. We have Kyungmi. Oh, the same name of myself. So, how many Korean company did you work with, and which company was your best customer? Have you ever worked with Korean company?
I have not worked directly with Korean companies. I've worked with companies from Japan, and it had a fantastic experience with that one company. Due to the nature of the agreements that we signed with some of our organizations, they're not always comfortable allowing us to use their name unless we're under protection of an NDA or CDA with other potential clients. Certainly anyone who is interested in knowing more about our experience with that Japanese client, I would be very pleased to sign an NDA and then have full disclosure. We do have permission with that client. What I will say is that client had not experienced any commercial success in North America prior.
Mm-hmm.
And they actually were looking at Canada as a market entry for all of North America, and to be able to evaluate their capabilities in moving into the North American market from a commercial perspective. They saw the value in coming to Canada from the perspective of learning more about how their product might be perceived and used both in a Europe-style market as well as an American-style market. And Canada presented a fantastic opportunity for them to do that. And so I was brought in to help assist with formation of the overarching strategic plan to begin with. They decided to enter Canada with a independent subsidiary, which they set up for themselves. I assisted them along with my team to ensure that they had the best regulatory plan possible, the best market access and reimbursement plan possible. We also helped to create a tactical plan for the strategy that was selected. And ultimately, we helped them in the hiring of their staff. And although I acted as the temporary general manager for a short period of time, I was very active working with the senior leadership to help hire the ultimate general manager for Canada and two of the most key employees that they brought in to begin the process of strategically resourcing from a human resource perspective the organization. So my experience with in the Asia Pacific market is not that great, but I do have a fantastic client experience with a Japanese company, and I would certainly look to be enjoying an equally exciting and profitable experience with a Korean client.
Okay, thank you very much for that answer. Well, actually, Yoon Ha-jo asked that question, and following, Minnie Gang also was curious about the success story of a Korean company, but you mentioned that you never worked with a Korean company. But for those of you who just watch it, this, presentation in Korea, maybe, Accelerate Canada, the first client in Korea. So if you are interested, please visit our website, BioKorea 2020 and Business Forum, and you can definitely have some website's address and contact number, email address to Accelerate Canada. Okay, thank you very much for your presentation and answer. I have to say goodbye.
Thank you. Enjoy the rest of the conference, and again, thank you so much for the opportunity.
Thank you. I hope to see you next year in Korea. Thank you.
Yes. Cheers. Bye-bye.
Bye. Okay, thank you very much. Our viewers are getting bigger and bigger. So again, don't be shy to leave any comments and questions. Okay, the next company I'd like to introduce is Vitro Imaging System. And this company is an innovative Belgian company developing smart quantitative microscopy solutions for life science applications. And Vitro technology offers machine learning, label-free, non-invasive tools for real-time 3D imaging of living cells. And this time around, Mr. Pascal Chabonnel here.
Hello.
Hi.
Yeah, can you see? Hello.
Oh,
This
sorry, Miss Pascal Chabonnel. Sorry about that.
Yes.
Okay. Oh, so we feel like we are traveling all around the world, from Australia, to Canada, and finally I see you here in Belgium.
That's right.
Right. So say hello to our viewers. Our viewers are getting bigger and bigger.
Hello. Good morning. Good afternoon to all of you. It's a pleasure to be here today. Thank you.
Thank you. Okay. Oh, I cannot really see you, but now I'm seeing your closet, I guess. Can you just change the camera? Can you just move your camera to you?
Yes, it is on me. How should I switch
Please
camera?
turn on the, yes, there we go. Now you are.
Okay.
Please begin your presentation.
Okay, thank you very much. And thank you again for the opportunity to present my company, Ovisio, indeed, its technology. So Ovisio Birds was strongly motivated by the need of a big pharma. At the time, it was 10 years ago, to have a system to really monitor their cells in a real time. So we have developed that solution for 3 years. And this is how our first microscope, the Island F, has hit the market at that time. So our technology is about 3D holography to monitor cells in a real time and without the use of any label, any dye. We combine our system with machine learning, AI, to really go deeper into the understanding of the cells. So in short, we provide image-based sensor to better understand, optimize, and control any biological processes. Next slide, please. So we partner with pharma company, biotech, and university. We historically address the field of vaccine development. So that's why you see Novavax, Sangamo, BI in the US, GSK in Belgium. And we enter more recently for the last 3 years into the field of cell and gene therapy with stem cells application with SunBio, Digenix in the US. And next slide, please. And we announced recently a strategic partnership with Celgene BMS, who has deployed our technology into their manufacturing network. And they're going to start the first in-human clinical trial this summer with the use of our device, the GMP compliant version. Next slide, please. So how do we interface into a process, an existing process? So this is an illustration of a CAR-T development process workflow. And where we bring value, it's at the stage, first at the stage of the expansion of the cells of the product. So you can see here a representation of our microscope, the I-line F, which is connected to this culture in suspension. And we provide also a complementary solution, which is the camera, the QMOD, the small camera that you can plug on a standard inverted microscope to monitor the product either upstream at the formulation stage or downstream if you want to control the quality aspect of your product or any other element that would be added in the culture, such as beads, for example, that people use for the activation steps. Next slide, please. So I'd like to see in motion, to show you in motion, the I-line F. So that's going to be the next slide in the video. So you have to play. So you should play. So this is how it works. You plug the cartridge into the microscope. The cells are circulating through inlet-outlet tubing, pump out from the bioreactor thanks to a pump that we're sliding here. And when the cells comes into the flow cell chamber and returning into the culture, this is where we're taking images and provides with the analytics per single cell, giving the density, the variability in a continuous way. So the system comes with machine learning, with a computer, which is install our software that we have developed in-house, OS1, where you will have a constant view on your cells right in the middle, and with the metrics associated at the right side, either in numeric or graphical mode, and you can customize this interface at your needs. So next slide, please. So the main feature, you have to press again, yeah, thank you. The main feature of the product is then it's all automated. You don't need to sample anymore your cells. You don't need to add any dye, so that which remove a lot of manual operation and related cost, of course. The system is digital combined with machine learning. And I'd like to mention that the next generation of the product will be CGMP by this summer. Next slide, please. We are quite versatile in terms of connectivity. So from small scale bioreactor to larger scales or wave bags for people developing cell therapy. The connection is done in an aseptic way, thanks to the fluidic part that is symbolized in the middle, which is the BioConnect that we have developed in-house also. The aseptic connection is done either by welding or by adding Lurelock connection to existing port of your bioreactor. Next slide, please. So the technology behind. So I'm not going to enter into the detail. It's just to explain why we don't use any dye. So because in fact we use the light and the capacity of a cell being a transparent object to refract the light. So when the light source will come, a living cell will generate a nice peak of light that we will measure. In fact, we will measure the difference between the light beam passing to the cells and a reference beam not passing to the cells, so more direct. So we will capture this delay onto a camera to allow what we call a 3D fingerprint of the cells. We reconstruct the 3D having the volume dimension in addition. And this is a first illustration of how we can discriminate between living and dead cells. You will see that the living cell is generating a nice sharp cone of light, whereas the dead cells have a pretty much spread signal. So this is a first assessment of viability. And if you click for the next slide, please. Combined with machine learning, we can measure up to 30 attributes per single cells. So it could be really linked to the morphology of the cells, so the diameter, the shape, the elongation shape. It could be linked to the optical characteristic of the cells, or very much linked to the kind of images that we've taken, which is phase or intensity image, and these are more linked to the texture of this image. So this is an illustration here of some of the parameters that we track. Next slide, please. And on the top of those parameters, we select, in fact, the most relevant, the most robust one, to develop the proper algorithm that the people will need to track their culture. So the more standard one and the more mandatory is, of course, density, viability. So this is here an illustration of how our system deliver the image and identify by having learned what is a living, what is a dead cells, what is a cluster. So we can count even the number of cells, let's say up to 10 to 20 cells, depending really on their size. So viability counting, so this is an illustration for CAR-T, could be done for TCR, T-cell, NK, and other therapeutic product. We can also identify or discriminate the cells versus other elements. I mentioned the beads. So you can see here that the beads have really morphological fingerprint, which is really different. It's round, small, black, compared to a normal cells, which is plotted in green in our system. And when the cells are entering into a morphological changes, let's say a differentiation or an activation shape, as you can see here, the elongation is really changing. The diameter is changing also, so that we can track giving a proper percentage of activated cells along the culture. So this is a feature that are more requested, needed for cell therapy. For the vaccine field, people are really much interested to follow the infectivity rate. So when the cells become infected by a virus, this is an illustration at the bottom as left side, where you see again that an infected cells behave very much differently from a normal cells. The granularity, the membrane integrity is very different, so that we can spot and give a proper, again, percentage of infected rate. And I will end up at the right side for cells in adherence. The confluency is a key matrix, and you have some illustration here on some cells. And should it be directly on the supervessel or on a microcarrier, we can estimate the coverage, the confluency on the microcarrier. Next slide, please. So here, to conclude, I'd like to show a concrete example of how our technology could be implemented into a pharma site. So this is the slide that BMS presented last week at the ASCGT Congress, virtual Congress last week. And they've presented basically their processes for developing T-cell and especially CAR-T. So they presented that before implementing our device, they had a lot of steps within their process, and many of the steps were manual. So the manual steps are in yellow, whereas some steps were already automated, and it is represented here in green. Next slide, please. And when they implemented our device, as you can see on the lab, so you recognize the align F connected to the culture, then they have very much streamlined their process. There is less step. It's 100% automated. There is no more manual steps. And they remove completely the biosafety risk, having a closed system. Next slide, please. And in addition to this improvement in terms of process, which is already great, they showed that in terms of yield performance, so they illustrated the viable cell concentration, VCC, along the time with our system automation, which is in pink, the curve in pink, compared to manual process, which is in blue, the blue curve, and it's almost 50% of gain in terms of yield performance. So it was really a great demonstration on what the value our system is bringing. So this is really the kind of partnership we would like to install with Korean companies. So that's why I have applied for the BioKorea. I think it could be a very good starting point. So I'd be very pleased to follow up with further discussion, and I'm open for any question at that time. Thank you very much.
Okay, thank you very much for your presentation, Mrs. Chabanel. Yeah, so you're looking for Korean partners, clients. Yes. So you never worked with a Korean company before. Well, did you have any meetings, specific meeting with a Korean company or Korean partners yet?
For Ovisio, no. Myself, I was working for BI, Ingelheim Animal Health Company, for the last 12 years, and I have a lot of interaction with Korean vaccine company at that time. So I really enjoy the technology, the spirit of Korean company. But that's why I was entering into Ovisio. I wanted to develop Asia market, and as a main focus for us.
So do you think the Korean, your product and your technology fits the Korean, market, Korean product well?
So I would answer that it's an international need. As soon as you prepare cells, either to develop a vaccine or a therapeutics, so it's really spread over the world, you have the need to reduce your cost. You have the need to control your process, make sure that everything will be okay in terms of safety and performance. So yes, I said that it would be a great chance to work together, at least to have a try and to see the benefit that we have already implemented at several sites.
Okay, thank you very much. It was Vitro Imaging System from Belgium. Thank you very much, Ms. Pascal Chabanel. I believe, as you mentioned, this will be the stepping stone for developing your original biotechnology and expansion to your business, Korea and Asian market. Thank you very much.
Thank you very much. Have a good day.
You too. Goodbye. Okay, so next, I'd like to introduce the company's Lab Genomics. And this company provides biotechnology and medical R&D services. And Lab Genomics' main business is clinical research, inclusive of in vitro diagnostics, biochemical. So this time around, Mr. Yang Jun-ho here for the presentation. So Mr. Yang, are you there? Hi.
I'm here. Do you hear me?
Yes, I can hear you. So Mr. Yang, would you please introduce your company?
Okay, this is Mitch Yang, and I'm working for Lab Genomics Overseas Sales Team as an assistant manager. And today I'm going to introduce about our COVID-19 RT-PCR kit. So mainly I'm going to speak Korean, and sometimes I'm going to use English. Okay. Yep. So our COVID-19 저희는 일단
Lab Genomics is dealing with the NDS and other molecular tests. And currently, we are focusing on the COVID-19 test kits. Currently, Corona is a pandemic hitting across the globe. So we are currently focusing on this kit. Can you pass to the next slide? Yes, I will briefly talk about this slide. Before explaining this slide, I would like to talk about the Corona kit. And before that, I would like to talk about PCR first. I believe that you're familiar with the PCR. It stands for polymerase chain reaction. And we use the thermocycler in order to amplify and then we read the result from the monitor of the PCR device. So PCR kit uses real-time RT-PCR technology. We call it as RT technology, and this RT refers to reverse transcriptions. So we extract RNA, and we use the our PCR device in order to amplify, and then we decide whether this patient is positive or not. So can you pass it to the next slide, please? Yes, I told you about the real-time RT-PCR, and then I would like to talk about the Corona kit right now. The marker for our kit is genome, RDRP gene, and E gene are our markers. And we use the two different types of genes in order to be used for the diagnostic kit for COVID-19. And can you pass it to the next slide? And this is the RT-PCR. Well, I will just pass this slide. And now, intended use. As I previously explained, I would like to talk about how to use our kit. You have to get the samples. You can get some swabs from your nose or the mouth, and you use those swabs and get the fluids from the nose or the mouth. And there is the reagent is added to it, and then this is how we collect samples. And we have the RNA extraction kit. We use this in order to extract RNA. And with this RNA, we use the real-time RT-PCR to make it to the DNA. And then with this DNA, we use the PCR device. We try to read the result in the monitor of the device so we can tell whether the patient result is negative or positive. Well, it is very difficult to see the slide from here, but I will just pass the slide. This is the application. We use the real-time RT-PCR technology for detection of SARS-CoV-2 coronavirus gene RDRP. So, as I said before, it is to detect RDRP and E. And the sensitivity is about 100%, and specificity is 100%, which means that we have accuracy of more than 90%. And we got these types of samples: sputum, trachea aspirate for the lower respiratory tract. It means that the fluids from the nose and the mouth. And for the time, it takes 6 hours. And the most part here is the PCR. And the PCR takes about 2.5 hours. So after these hours, we can read the result from the monitor of the device, and with a graph on the monitor, we can read the result to tell whether this patient is negative or positive to the virus. This is the PCR device. And I believe that you're familiar with the open system. When we develop our kit, we consider these two types of the thermocyclers, and we had tests with these two cyclers. So, and these were manufactured and developed in the US. And one of them is a real-time PCR CFX96, and the one on the right side is Thermo Fisher. This is a famous US company. This is so-called AB 7500. So these are the PCR device or the thermocyclers that we use for our kits. And you have to have these cyclers in order to carry out the whole diagnostic process. And this shows the regulatory clearance. We got the approval from the Korean Ministry of Food and Drug Safety. And also we got the EUA, CFS. And the most important part here is FDEUA. It means that we got the approval for the emergency use authorization, so we can sell this in the US market. And actually, we register in some countries, including Greece, Italy, and India, and in progress, Philippines. Yes, we are already registered in these countries. So we can sell these products in those countries. We'll have a Q&A session after the presentation. Next. This shows the kit components. I believe that you just have to read this. The most important part here is SA1 and SA2. These are the markers. SA1 detects RDRP gene, and SA2 detects E gene. The other companies ask us a question regarding engine. However, according to the recommendation of the WHO, we have to detect RDRP and E genes. And these are the two things that we are considering right now. So the rest of them are just control. It was designed and included to check whether the PCR is completely done. Next slide. This is the packaging of our kit. And the smallest unit of the package is 100 sets. So in the one box, there are 100 kits. In this small box, as you can see from this slide, the 7 agents are included. I'm sorry, 6 agents are included. And this shows how this box, what kind of component is included in the box. Next. Yeah, this is another picture that shows our packaging in the kit, and I think you can just refer to it. Next. Yeah, this is a kit box. As you can see, we wrote COVID-19 RT-PCR kit. It is an official name for the US market. Next. For the storage temperature, I think it is all same for the PCR. For the reagents, the minus 20 degrees is recommended. So in order to store these agents, you have to maintain a temperature of negative 20 degrees. And for the shelf life, it is 12 months from the date of manufacturing. I think it is all similar or same for all agents. And next page. And in order to deliver the product or kit to you, the packaging and the handling are really important. I'm not so sure whether you can see this in details or not, but still, depending on the units, we have different cases or different boxes. So in the strip-on box, we put the agents or the kits, and then we put the dry ice, and then we pack it, and we have case for 100 tests, 3,000 tests, 7,000 tests, and 20,000 tests. And the biggest box contains 20,000 test kits. So it has a different weights. We can provide this data to you if you need this information because the cost can be different according to the size of the packages. Next. These actually I briefly mentioned before. Well, this shows the whole process from sampling to the analysis of the results. I will just pass this slide. Next. And this shows how we get the sample. We use the swaps, and it's for your reference. Next. And I would like to talk about the second process of sample preparation. The most important part here is that there are PCR devices that you can use our kits with. One is from Thermo Fisher, and we have to extract the RNA, and there are devices to do that. And Cuagen, this is one big company, and you can get the extraction kit from Cuagen and Genol, and these all actually what we use when we develop this kit. So for the RNA extraction, you can use these kits. And can you pass 2 pages? This shows how we analyze the results. There is a monitor of the PCR device, and I believe that you can see some little graphs on the slide. After a small period of time, there is a point of RT, and if it is higher than the line of RT, it is positive. It is lower than the negative. So it is very easy to see whether the result is positive or not. Next. This is current production capabilities. Every week, we can produce up to 10,000 kits. And some people ask us about the stocks. And well, really, it really varies. It's actually from 1 million to 2 million. Next. And this shows the specification of the PCR device. Next. And this is also what I explained before. This is a list of the RNA extraction systems. I think you can just use it for your benefits. And that was the end of my presentations. We will now have the time for the Q&As.
Inquiry? Yeah, I'm done. Any question and inquiry, please ask me.
Thank you very much for your presentation. 발표 감사합니다.
네.
네. 자, 저희가 지금 채팅창을 보고 있는데요. 그래서 checking the live chat. 질문이 꽤 많이 들어왔습니다. 그래서 질문을 드리도록 하겠습니다.
We have a lot of questions listed. First of all,
your comments and questions. We have 이리엇, 보희킴, 민희강. Hello, thank you very much for tuning in. 첫 번째 질문은요. 먼저
the first question was regarding the FDA clearance. Yes.
We got FDA EUA authorization and also C certificate. We got both of certificate.
자 그리고요. diagnosis kit.
래피드 키트라 말씀하셔서 신속 진단 키트를 말씀하시는 것 같은데 일단 저희가 현재로서는
Currently, we are not producing rapid kits. We have a plan to develop it further. So you have a plan to develop it further, okay? So what about the technological development directions? What is the direction that you are taking? So NGS is what we are focusing on. That is one of our main businesses. And there is an NITP as well. In order to test the mothers with the babies to figure out whether the baby has a defect or not. And for the PCR, well, actually, I talk about the PCR kit today, and it is used for the COVID-19 and other diseases such as Zika and TB.
스웹이랑 RNA 추출 키트는 공급을 하고.
So do you supply the RNA extraction kit as well? Well, we are producing the PCR kit only, so RNA extraction kit is not really what we are supplying to. However, we can add that clause to the agreement. So what is the capacity of the productions for the whole week? How much is it? Every month, we can produce 1.5 million. So it is about 1.5 million. So it is a huge capacity, I think.
4 or 5 questions to lab genomics. Yeah, people are so interested in your company and technology. Thank you very much, Mr. Yang, for your wonderful presentation and Q&A session as well. 감사합니다.
Thank you. Yeah.
So, BioKorea Live Talk 2020, our viewers are with us. Thank you very much. So this program is to share pharmaceutical, bio, and healthcare companies that strategies as well as visions to investors, people related in this field through online live stream service. So viewers, please feel free to leave your comments and questions, and you can win some gifts. So now, let's dive into the company, Colby. Well, actually, we are in the halfway of today's presentation. We have a total of 8 companies' presentations ready. And this will be the 5th company, Colby. Yeah, sounds so cute, Colby. So this time around, Mr. Choi Chul-won will give us a detail. Hi, Mr. Choi. Are you there?
Hello.
Hello, can you just turn on the video? Yes, here. I can see you here. So,
Yeah.
how did you get the name for the company, Colby?
The Gulbi means that the honey lane in Korean.
Mhm.
Honey lane means that the rainy is a little bit is feel like the sweetie. So we think it's Gulbi is very good for everyone.
Okay, thank you for your
Thank you.
explanation,
Yeah.
Colby. Okay, and the Colby is a software company based on big data and machine learning, and the company is located in Daejeon City, right?
Yeah, right.
Oh, you are in Daejeon City.
Yeah. We stay in Daejeon and near the KBS.
Oh, okay.
Yeah.
Okay, so for those of viewers, if you visit to Daejeon City, just feel free to visit Colby. You're welcome, right?
Thank you.
Okay, so please begin your presentation from now on.
Okay. So hello. Yeah. Let me introduce the Gulbi first. So our today presentation contents is listed company overview and success story over quantum and core the competence. So the presentation is not ready. Okay. So about Gooby, our vision is Gooby vision is to find the core and value in the data and lead the world. In not just about the data-driven system development, through research to the connect customer and platform. I want to be the brand that want to deliver positive experience to the users. The next slide, please. The next slide, please. The next slide, please. Yeah. So we think is to the four category of the competence is very important. The first thing is the insight. Find the core, not just the side things. So, and the second is valuable. So we create the values to the people, and we focus on your data, and we try to solve your problem, and we also consider the trendy because this value of thing is represent the near the world, so we need the word. Next slide. So our corporate name is Scooby, and it's founded from the March 19, 2015, and we have 15 the employees. And the next slide, please. Yeah. So our story is starting to the 2015, and so the first we development the franchise marketing management system, and second development near-time stream using the FP MVPG 2016. So we working in the R&D and we make the web management and marketing for wholesale and retail on United States, and AI technology based on the intelligence, the learner assessment system, because that at that time the Isedo and the AlphaGo they matched to the Baduk, and we realized that the AI technology is gonna be very popular and impact on the worldwide. And the 2018 we starting to the convergence with the BT and IT system. So we developments or web-based RNA-seq genetic analysis tool, and 2019 we working in the public institute work, a big data center, and we work for this, the building the big data system. Next slide, please. So we have the many key customer partner already. For examples, the Korea University or Posedu or the GIST or Chungnam University like that. Next slide. So we during this journey we had the many awards as well. And we won the AI intelligence technology the competition. And next slide, please. So we have the Gooby has the capacity with the data science, machine learning, or web app service development. So we want to find the collaborator. We have the clinical research field or the development or the medical field. We want to convergence together with the health tech as well. Next slide, please. So our business vision is gonna be number one, HTIT service group. So we plan to do the Kostak IPO in 2024, and we gonna the machine learning and biotechnic company to be. So next slide, please. So we also getting hire the talented the people. And next slide, please. So our Group is incorporate with the many position with data science, biologists, medical doctors, bioinformatician, where machine learning developer, applied software developer, and system engineer, and as well as designer together. And next slide. Next slide, please. The next slide. Yeah. Next slide, please. Yeah. So we already we has many records from the working the business area, the many things. The field is categorized the four field is the first of all the bio and medical data analysis field, and the machine learning and artificial intelligence field, and data collection and the manufacturing field, and service platform development field. Based on this field, we working with many the public institute and private company or many and during this collaboration we developed the system related AI and big data system. Next slide, please. For examples, we make the online test near browse supervision system, and it is the next slide, please. And then we making the support for community care system, and this is comprehensive health requirement diagnosis system. It's customized the care management system. And next slide, please. Not only the for medical the field, we also develop some the land price the value system based on the random price wrapper and NLP rich and KEN algorithm. We also registers patent as well. The next slide, please. So we have the many experience with the big data system development. In case of the Forest Welfare Promotion Agency, we developed the big data center and with Bombsoft, we consult with them the making big data with the child care planner system as well. The next slide, please. So we nearly focus on the quantum. This is our own the solution, not working with others. So next slide, please. So we do the many R&D now, but we think is developing the HT with IT convergence. We make more the step-by-step R&D approach strategies. The first step, so we do the genetic analysis system, and the second, the RNA. We now do the RNA natural language processing based on the RNA the papers. And the third thing is we do the protein modeling, new drug development through the platform. Based on the results of R&D, we want to try to working with the hospital or medical now. So we discuss with some hospital to working with the medical big data field. The next slide, please. The quantum is AI-based RNA-seq analysis. So according to this system, you can the analysis with the RNA without any delay, because it's right away you can see the results after you upload the prosecute the files. Next slide, please. Not just to the analysis, the genome data, the AI system may provide the paper information with natural language analysis techniques, because many papers is publishes about the 50,000 annually, but you cannot read every papers. So it helps to summarize which the subject is very important or not, according to your base, your research. Next slide, please. So the quantum is compromised with the biotech and machine learning together. The biotech field is compromised the engineers analysis pipeline and UI UX, the gadget analysis tool together, and machine learning part is compromised with intelligence pathway database and artificial intelligence analysis of dissertation together. Based on this technology, this provide the clinical relevance, atomic analysis, and machine learning based disease analysis, and gives gene pathway or disease correlation information as well. So you can see the gene pathway disease, their relationship together with the intelligence database. Next slide, please. The quantum is automated system for establishment the assay information based on the natural language process. We download from the NCBI, the PubMed database, because they provide the public DB, and based on this abstract, we process the data and filtering out the unnecessary data, and based on the syntax net or based the BERT, the natural language algorithm, and it's not enough for syntax net or BERT enough, because their results is not fit with our purpose. So we make the parser and we more divide them, and this store X, Y, Z DB, and we making the natural language based database now. Next slide, please. Next slide, please. Yeah. So we have very strong core competence with big data. Next slide, please. And data science. Next slide. Yeah, next slide, please. The machine learning, the technology, and so we want to be the collaborator or partner with the medical field or the biotechnical field together, because our company is very strong ability to based on the IT technology now. Next slide, please. Okay. Thank you for presentation. If you have any question or more detail about our company information, please give email or call the phone. So thank you for listening and pay attention to my speaking. Thank you.
Thank you, Mr. Choi, for your wonderful presentation. It was really, helpful to understand what you're doing, what your product, your technology. So we have listened to Corby. Thank you very much.
Thank you.
So we've got about 2 questions. I'm checking now on our live chat. 자, 질문이 2개가 나왔는데요.
So we have 2 questions here.
Which of bio or IT characteristic is more important? Bio or IT? 자, 바이오와 IT 특성 중에 어떤 게 더 비중이 높은 건지, 회사가 어떤 비중을 더 많이 갖고 있는지 궁금합니다.
네, 저희는 BT에서 출발한.
Okay, so basically we are not a company that started in BT biotechnology, like our members or our capacity they're all around IT. So I would say that comparing to those who work on the new drug development platform, they have to work with other IT company to resolve these IT related parts. But our company, so to speak, at Gulbi, I personally am responsible of the bio part. I've graduated from KAIST, so I do have some expertise, and that's what I provide. But most of the members are IT experts. So I believe that this kind of structure embody lots of strength. For BT, for example, of course, we can advance and we can, for example, acquire the intellectual property. So fast developing of technology can be a weakness for the BT-centered company. But as we are a company that combines these two, I think we are able to improve that part and resolve that challenges. Thank you for your answer. And the second question is, so what are your directions of the technological development? It is indeed a wide question, but yeah, so our directions for technological development, of course, all companies working on AI or BTOIT, I think it is shared by all, but their goal is the new drug development. So for making it possible, what would be the important factor? I believe we all share the same final goal, but maybe we have different paths. And these are the questions that maybe some other companies also have. And us, we plan to achieve the goal through the IT technology. So back in 2018, we're designated the local IT company of the city. So we have completed the convergence technology, and with our natural language process technology, as I explained, we can extract some of the useful information from the papers using the new technology. And starting 2019, we'll be introducing the machine learning and artificial intelligence technology, so that when we carry out analysis, the genes will be the target genes. And later on, atomized, or just like other company, maybe with this modeling, we will be able to advance further. Thank you so much. Thank you. I believe that you do have very specific plans ahead, and thank you very much for your presentation and for your answers. So any final comments that you may have? Okay, so if I leave you with my final thought, at Gulbi, our vision and motto is my happiness, our happiness, and the global happiness. And that means that our happiness and my happiness should not be the ultimate goal. Our ultimate goal should be the global happiness and happiness that is shared by all. So it is not just about our interest, our company's interest, but also we take care of our members' welfare, members' company. So basically, I'm trying to make the company as the one that can embrace everybody's happiness. So please contact us if you're interested. So just like your name, I believe that your company is very socially responsible one that is not only interested in individual happiness, but also work so hard for everybody's happiness. That was an impressive final comment. Thank you so much.
지금 멋진 분들 그리고
And I think there are a lot of people are interested in the company Gobi. And if you're looking for a partner with Gobi, please don't hesitate to contact him. It was Gobi.
네.
네. 자, 이제 많은 분들이 계속해서
Well, actually, many people are participating in our live talk.
bigger. And we'll still waiting for your feedback, your comments, your questions. So just feel free to leave your message. 네, 고맙습니다. So that was 4B. I think the company is looking forward to tomorrow more than today. Thank you very much once again what you said. Moving on to the next company is Eurofins Discovery. And this company offers one strong foundation for successful drug discovery. And Eurofins Discovery offers you a complete single source solution for drug discovery products and services. So I think there are two companies in this segment. So I'd like to call our presenter, Mr. Choi Chul-won. Oh, I'm sorry about that. Mr. Kim Ju-young. Hi, Mr. Kim Ju-young. Hello, can you hear me?
네, 여보세요?
네, 여보세요. 안녕하세요.
Good afternoon.
Oh, Kim Ju-young. All right. Okay, you're on here.
네,
Hello, good to see you.
네.
네, 안녕하세요.
Good afternoon.
Two companies on my list. So, Giant Laboratory and Eurofins Discovery. So, how are they related? 자, 두 개 회사가 저희 리스트에 지금 함께 올라와.
What are the relationship between the two companies listed in the table, the Eurofins Discovery and the Giant Lab? Giant Lab is a Korean company. So these two companies are basically doing the same. Yes. So you can start the presentation. Please send a lot of comments and the question, the live talk.
In the presentation.
Good afternoon. I am Kim Jo-young from the Nature Lab. I'm sorry, because of the audience issue, the interpretation cannot be provided right now. I'm sorry. Once again, the interpretation cannot be provided.
다음 네.
Eurofins Discovery has a bio platforms, and it acquired the premier niche of CROs, and also I'm sorry, because of the audio issue, the interpretation cannot be provided. Once again, I'm sorry. The interpretation cannot be provided because of the audio issue.
네. 지금 잘 안 들린다고 하셔서
네, 김주영 차장님 잠시만요. 저희가 지금 사운드 체크를 좀 다시 하도록 하겠습니다. 약간 울림이 있어서 저희가 기술적으로 다시 한 번만 체크를 하고요. 발표를 이어가도록 하겠습니다. 네. 잠시 떠나지는 마시고요. 저희가 금방 바로 복구를 할 수 있을 것 같으니까요. 차장님 죄송합니다. 불편 끼쳐드려서.
네, 지금 혹시 괜찮은가요?
Yes, I think it's working now. It's all resolved. I'm sorry there was the audio issue. I think it is okay now. Yes. Yeah, I can hear you now. I'm sorry, can you start all over again? Eurofins Discovery is the inventor of biological platforms, and it also acquired the Discover Access Arab, and it's a leader in this industry. Eurofins has about 7,500 validated assays. Also, they are the PIs in the lab with an experience of more than 10 years. And for each discovery stage, we support with our utmost effort. Next. Eurofins was established in France in 1987, and it encompasses biopharmaceutical, clinical diagnostic, environmental, and food products. And actually, it provides analysis for all of these products. And they are focusing on the biopharmaceutical analysis. Next. Eurofins has branches in the Taiwan, Europe, and also about 14,000 projects are being conducted every year. So if you want any services, we can provide the nation customized or the need customized support. Next. Eurofins is dealing with a whole process from early discovery to the clinical services. If you take a look at the parts, including chemistry, in vitro, phenotyping, ADME, enviro, and products. Also, we provide the products in the form of the cell lines. So if you have a specific stage in mind, you can select the stage and the services that you want. There are 4 different stages: early discovery, hit finding, lead optimization, and clinicals. So you can choose the different stage or experiment to have the specific services. Next. In vitro pharmacology services. We selected potency, selectivity, safety, ADME, and toxicity, efficacy. Well, all of these can be tested, so these satisfy the needs of the pharmaceutical companies. In particular, Eurofins, cyclodextrin, ion channel, and well, actually, we have studied target classes such as GPCR kinases and ion channels. And we already have the several gold standard panels. And we have a lot of other popular services, and many customers are utilizing our services as well. Next. Eurofins are supplying a variety of assays, as you can see from this slide. GPCRs. We have more than 2,750 assays. We have more than 1,000 kinases and 400 ion channels and more than 178 NHRs and more than 50 of IONC targets. And we have a lot of services regarding different types of assay types for the GPCRs, PMP, calcium. We have different assay types. And for the kinases, binding activity, cell-based assay, and the KD assays are all provided through us. And for the ion, binding, Pechiclampu, MEA, flipper assay can be supplied in order to meet the needs of the customers who can select the type of the assay. And in addition to that, protease, costopise, PD, ligase, brodomain, and tubex, all targets that we can provide to all of you. Next. Well, this shows the case study of the flowchart of the HTS. First of all, we designed the assays and decide how we are going to do it. And then we are going to miniaturize assays, and then we're going to optimize the assay conditions. And we conduct industrialization of the assays to establish the platform. And then we have pre-screening for the drug screening. And on the right side, you can see the table. About 100,000 compounds can be screened, and it is all finished with 6 days with average hit rate of 0.43%. So, Eurofins provides the optimized services and platforms. Next. These shows the SAR and the optimizations. These are the case studies. At this level, many drugs related EC50, IC50, and MOA are defined through binding functional assays. We conduct multi-site screenings in order to define all of them. So if you're developing some drug or the compounds, we can deliver the result in a swift manner. And we can provide the customers the services according to the needs of their customers or clients. Next. Now, ADME and toxicology services. For the safety panels, we have the global gold standard. Eurofins safety is a non-GLP services. And for the ADME, we provide tailored to the services, and you can choose from 2 panels, and these assays are of non-GLP. In vivo DMPK. On the right side, you can see the kit compounds. You can select the candidates, and you can have the services at the stage you defined or you picked. All of these customized services can be provided from the Eurofins. Next. From 2000, Eurofins has been conducting phenotyping drug discovery, and we have been the leading this industry with more than 500 clients. We have been providing the customer services. You have the human primary cells, and it is so complicated process. However, we can test the efficacy of the drug against the humans. So we provide services for human cell-based disease systems for physiological relevant assay data, and we use the database in order to predict the mechanism of the drugs. So you can select the benchmark compounds, and we can compare your drug to the benchmark drug. And recently there was an issue regarding cytokine, and we provide services regarding the immunology. And anti-cancer agent screening, we can complete 300 screening process for the cancer cells, and we can use the genomic data and other results of the analysis. So if you have a specific phenotyping services in mind, we can provide the services in a variety of forms. Next, in vivo safety and efficacy. We already conducted experiments for over 25 years in Taiwan. And for each efficacy model, you can choose disease, and you can have a CNS oncology or allergy related data can be collected from our platform. So if you are interested in a specific target disease and you want to use the in vivo platform, we can provide that as well. And we have the customized in vivo experiments as well. So if you have something in mind and you can have some items to be considered in the design process, we can take consideration of them. Next. Eurofins have tools to test the reaction of the drugs in the lab. So we have different stages of the cell lines. We have the agents. I'm sorry, the audience. I'm sorry, the interpretation cannot be provided because of the audio issues. I'm sorry, the interpretation cannot be provided right now because of the audio issues. And the cell lines can be optimized And for the biosimilars, we can use the drugs. And for these drugs, we use the biotherapeutic QC. So if you have interested in the research regarding biosimilars, we can provide you biosimilar assay ready kits. Next. Lastly, Eurofins is trying to provide the customer services in the era of the COVID-19. So these are the platforms that we have that can be used for the COVID-19 in order to respond to this disease in a swift manner. And Eurofins is going to put their utmost effort to provide those services. And if you have a need to have this R&D or test, you can contact us. Lastly, Eurofins has experience of more than 40 years. We earn the trust of the clients and the customers, and we have a flexibility and we have adaptability to respond to the needs. And in terms of the quality, we can provide the best quality analysis and the results and the products, and I'm sure that we can be really helpful. Thank you. That was the end of the presentation, and it is my honor to take part in this BioCorea 2020. And currently, if you have interested in the services that we provide, please contact us to the Chaeyeon Lab or Eurofins Discovery, and I can provide you more detailed information regarding the services that we provide. Thank you.
네, 감사합니다. 발표 잘.
Thank you very much. I think you have a lot of and a variety of the services that can be offered. And we are here with us 2020 BioKorea Live Talk. And we have the virtual exhibit and there are business forums. And are you taking part in other parts of the 2020 BioKorea? Yes, we have the VR exhibit. You have a variety of the services. So if you're interested in the company, please visit the virtual exhibit on the homepage of the 2020 BioKorea, and we have the email addresses and contact numbers. So if you're interested in, please don't hesitate to contact them. There is a high interest in the COVID-19. And it is a hot issue right now. So Eurofins has this platform to respond to the COVID-19. So I think it is very interesting. So I hope that a lot of you have interest in the company and contact them. Thank you very much. It was Kim Joo-young. Thank you.
고맙습니다.
네, 감사합니다.
네, 고맙습니다. 자, moving on to the next presentation. Our seventh company is GenScript, and Mr. Edward Wang is now ready for the presentation. GenScript Biotech Corporation is a global biotech company, and the company's mission is to make the human and nature healthier through biotechnology. And GenScript Biotech Corporation is a leading life science research in application services and product provider. So with a short introduction, I'm very wondering about this company. So hi, Mr. Edward Wang.
Hello.
Hello,
Hi.
Mr. Wang. Hi.
Can you hear me clearly?
Yeah, I can hear you, and I'm
Very
so
nice.
good to meet you there. How are you?
Good. How are you?
Good, pretty good. So, have you ever participated in BioKorea? It's annual international event in Korea.
Yes, I actually did a webinar just a couple of weeks ago talking about the COVID-19. And then I'm honored to be invited again to participate in this BioKorea.
Okay, next year, we'll hopefully to see you here in Korea, the venue. And say hello to our viewers. We have many viewers here. Hello.
Hello, everyone. Hello. Nice to meet you guys.
Good. Good to meet you here. So please start your presentation.
Okay. So thank you for the kind introduction and good day, everyone. It's a great pleasure for me to have such a wonderful opportunity to give a presentation on this BioKorea webinar 2020, as I mentioned earlier. So in this webinar, I will be sharing an overview of COVID-19 and also its latest information. Next slide, please. Yep. Sorry, next slide. Okay. It's the content, right? Okay. So let me guide you through the contents of my presentation today. So I will give an overview about our company, followed by a brief introduction about the SARS-CoV-2 virus and the detection methods provided by GenScript. And lastly, to briefly touch on the services and products provided by GenScript to help the researcher in vaccine development. So, okay. I would like to take a minute or two to briefly introduce our company and the milestones that we have achieved throughout these 18 years. Our company established in 2002 in New Jersey, United States, and was the first company to commercialize gene synthesis. In 2004, we established Research and Production Center in Nanjing and expanded our offering to protein and antibody as well as the peptide services. After over 10 years of development, we won the CRO Leadership Award in 2014. GenScript was listed on the Hong Kong Stock Exchange in the year of 2015. In 2017, the Legend Biotech, which is a subsidiary of GenScript Biotech Corporation, that developed the innovative CAR T-cell immunotherapy, had collaborated with Janssen to jointly develop and commercialize the product. And in the same year, the China FDA accepted the IND application by Legend Biotech. Next slide, please. So backed by the cutting-edge proprietary technology, GenScript Biotech Corporation is a leading life science research and application service and product provider. Currently, there are more than 100 patents under our belt, and more than 300 patent applications are still under processing. To date, we serve more than 110,000 customers from over 160 countries, and regions around the world have used GenScript Premier, convenient and reliable products and service. Additionally, GenScript has more than 3,700 of employees globally, with over 37% of employees holding a master's degree and above, in order to help in providing the consultation to our clients in their study. Next slide, please. So, based on the leading gene and protein synthesis, the group has established 4 major platforms, including a leading contracted research organization, which is known as a CRO platform, to provide a one-stop solution to global research. Second, a contract development and manufacturing organization, which is known as the CDMO platform. And third, an industrial synthetic products platform. And fourth, an integrated global cell therapy platform. So now, given the rapid spread of the SARS-CoV-2 virus globally, I am going to discuss about the COVID-19 solution that's provided by GenScript. Next slide, please. Okay. So when we look back at the history, coronavirus had caused a few epidemics in the recent years. So taking the SARS as an example, it was first reported in Guangdong in the end of 2002 and had infected more than 8,000 people globally and killed more than 700 people. And in 2012, the MERS virus was first reported in Saudi Arabia. And since then, about 2,500 MERS cases have been identified across 27 countries and have killed more than 800 people. And now, we are facing the threat from the SARS-CoV-2 virus that resulted in the COVID-19 disease. And it was first reported in Wuhan in the end of 2019 and had now spread to worldwide. As of 17th of May, the SARS-CoV-2 virus had infected more than 4.17 million people worldwide. and killed more than 315,000 people. And the most worrying bit is this number is still increasing. On 11th of March, the WHO declared the COVID-19 outbreak as pandemic. Next slide, please. So here is a picture of the coronavirus that released by the National Institute of Allergy and Infectious Disease. So the coronaviruses is actually a large group of viruses. They are the positive sense single-stranded RNA viruses. The genetic materials are surrounded by the envelope protein with a spike protein. There are different types of the coronaviruses that cause the respiratory diseases and the gastrointestinal diseases. The respiratory diseases can range from the common cold to pneumonia, but very often these symptoms are mild. However, some can cause the severe symptoms, such as the SARS, the MERS, and now the COVID-19. Next slide, please. So, how coronaviruses hijack our cells? The coronaviruses spike protein can mediate the infection by binding to the targeted specific receptor known as the angiotensin converting enzyme 2, or we call it ACE2 receptor with high binding affinity. The S protein contains of predicated S1 and S2 subunits. The S1 subunit contains of the receptor binding domain, which is known as the RBD, and the non-RBD region. And the S2 subunit has been found to mediate the membrane fusion between the virus and our cell. Next slide, please. So, based on our current understanding to SARS-CoV-2 virus, GenScript has offered a total solution that covers the diagnostic field, including the nucleic acid-based assay and the serological assay, and the services and products that help our researchers in vaccine and drug development. Next slide, please. So, given the success in sequencing the viral genome, both the WHO and CDC have released the sequences of the primers and probes that targeted 4 different locations in RD-RP gene, E gene, N gene, and OF1AB gene. And based on this, GenScript not only helped to develop the primers and probes, but also to develop our own qPCR kit that include the one-step, one-plex detection assay to target the 4 different locations as I mentioned earlier, and a one-step, 2-plex detection assay with the combination of OF1AB gene, N gene, and the E gene and RD-RP gene. Additionally, we also provide the positive control plasmid for detection. Next slide, please. Sorry, next slide. Okay. So here I would like to take this opportunity to introduce our serological assays, including the ELISA kit, as well as the newly launched surrogate virus neutralization kit, or we call it a SVNT kit, which is a collaboration project between GenScript and Professor Wang Lingfa from Duke NUS in Singapore. As we all know that the ELISA is a plate-based assay that relies on the antibodies to recognize the antigen. Based on this, we have developed 2 different types of ELISA detection kits. In the first type of the ELISA kit, the plate is pre-coated with S1 RBD that will be recognized by the human IgM and IgG binding antibodies in the serum. In the second type of the ELISA kit, the plate is pre-coated with a fusion protein between the nucleocapsid protein and RBD that can be recognized by the human IgM, IgG, and IgA binding antibodies in the serum. So after washing to remove the unbound antibody, it will then be incubated with the secondary antibody and conjugated with the enzyme. The assay is done by assessing the enzyme activity to incubate with the substrate. And now, I would like to switch gears a little bit to single out and discuss more in details about our newly developed SVNT kit. Next slide, please. Thank you. Even though there are numerous serological test kits available in the market, there are questions still remain to be answered. First, we want to know the herd immunity and interested in finding out when a person can go back to work, which we call an immunity passport. We also want to know how long can the immunity in the recovered patient last for after the infection. Another thing is the development of convalescent plasma for therapy. We have so many patients being infected to recover. When they donate their plasma, we can develop a passive immune therapy. But how can you determine one of the recovered patients is better than the other, or whether they are sufficiently effective? We know that there are a couple of vaccines under the clinical trial at the moment, and very soon we will come to the point to ask which of the vaccine can give a better efficacy. Researchers are still in the search for the natural reservoir or intermediate host that involves in the virus transmission. And lastly, to monitor the possibility of reverse zoonotic transmission. There are scientific reports indicated that when the human is in close contact with the animal, such as the dog, the cat, the zookeepers who are taking care of the animals in the zoo. Now there is a possibility that we humans can infect these animals. So the first 5 points, which highlighted in the red, will require us to know the neutralizing antibody. But unfortunately, none of the commercial kits these days that are available in the market right now can tell us the answer because they only measure the binding antibody. And to look for the natural reservoir or intermediate host, the researchers will need to conduct the animal testing. So we need to develop a serology test that is species independent. Yet again, none of the serology kits available right now can do that. So with this newly developed kit, it can be used not only to the human, but also to the animals, including the pets in your house. Next slide, please. Here I am going to show you how this kit actually works. This kit works differently from the usual ELISA kit in principle. As mentioned in my previous slide earlier, that the entry mechanism of the SARS-CoV-2 virus to the human cell is through the binding of the spike protein to the targeted specific receptor known as the ACE2 receptor. So when our immune system starts producing the neutralizing antibody, what these antibodies do is to disrupt the binding between the spike protein and the ACE2 receptor by blocking the spike protein. The conventional virus neutralizing test will rely on the use of the virus in the study. And this is very dangerous. You need to have a highest level of the laboratory setup to handle such a contagious virus. However, with our SDNT kit, rather than using the real virus, this assay utilizes only the RBD that are conjugated with the HRB. So as I introduced, the receptor binding domain of the virus plays a major role in binding to the ACE2 receptor. So in this assay, we will be measuring the inhibition effect of the binding due to the presence of the neutralizing antibodies that present in the serum of the recovered patient that block the binding between the RBD and ACE2 receptor. The study has shown that not all the patients will carry the neutralizing antibodies even after the recovery. So with this assay, it could help us to understand if we carry the immune passport when going back to work post-COVID-19. Next slide, please. Here I show you the inhibition effect of the assay in the presence of the neutralization antibodies in the serum. The x-axis indicates the serum in different dilution, and the y-axis indicated the inhibition percentage. In negative control, which were both healthy patients, the results showed no inhibition effect. But the serum from the COVID-19 patients with the presence of neutralizing antibodies, we could clearly see the inhibition effect and a reduction in the effect after the dilution. Next slide, please. So as I mentioned, what's unique about this assay is it is species independent. To prove that, Professor Wang Lingfa had conducted the assay on the rabbit and the ferret, and both show that inhibition effect due to the presence of neutralizing antibodies after the infection. Now that could mean that this assay is suitable to be used on the animal. Next slide, please. The author had also compared the SVNT assay with the normal ELISA assay that measured the human IgM IgG. The ELISA assay demonstrated a discrepancy result in the IgM and IgG antibodies as shown in the figure. And this was expected because we know that the human IgM and IgG are produced at different states after the onset of the disease. Usually, the IgM will produce first, followed by the IgG. In contrast, SVNT had no such issue due to the reason that this assay was to determine the inhibition effect due to the presence of the neutralizing antibody. Therefore, our results show that the reading of SVNT kit was pretty constant. Next slide, please. Right. Additionally, when tested on the patients across different regions, this assay demonstrated 100% of the specificity and a near 100% of the sensitivity. Next slide, please. So, the advantages of using this SVNT kit are its high specificity and sensitivity. It is safe to be conducted without the use of the real virus. The assay only takes less than 1 hour to complete. It can be used for both low and high throughput screening and even for the automated purpose. This assay is species independent and can be used for both humans and animals. And lastly, what I would like to emphasize here is this assay has already received the approval from the Health Science Authority in Singapore and the CE mark, which means that this kit is ready to be used for the clinical diagnosis. Next slide, please. Next slide. Okay, thank you. While the researchers are currently in a race to develop the vaccine and therapy for COVID-19, GenScript has spared no effort to help the researcher to speed up their study with our in-house technology. But due to the time constraint, I will only briefly touch on the services and products that are offered by GenScript, and you are always welcome to contact me or our sales manager in Korea to discuss more in details after the webinar. To help our clients to discover the suitable neutralization antibody candidates, GenScript can help our client to test their neutralizing activity test through our pseudovirus neutralization assay service. Additionally, we are offering the ACE2 Express assay cell line and the pseudovirus reagent for the researchers to conduct the assay on their own. We can also help our clients to speed up the antibody discovery process by utilizing our in-house cutting-edge technologies, such as the Bacon platform. And we already have to control antibodies against the nucleocapsid proteins and the spike proteins of the COVID-19 that can be used as a control in the study. Next slide, please. We know that the currently used vaccine candidates are mainly DNA-based, mRNA-based, recombinant proteins or peptides. GenScript offers a wide range of services and products that cover all of these areas, including the peptide synthesis and peptide libraries to generate the possible candidates for the study, a research grade to a GMP grade of plasmids that are readily to be delivered to the cells, and last but not least, with our advanced synthesis technology, we can help our client produce the recombinant and subunits as listed in my slide. Next slide, please. So before I finish my presentation today, here I show you a couple of classic examples about the partnerships that GenScript has formed, not only with the academic researchers such as the Professor Wang Lingfa in order to help them to develop the assay, but also the biopharma companies such as the Novavax,
So there's a kind of technical issues. We apologize for your inconvenience. So it takes a little time to make it perfect. So please wait a moment and the presentation will be starting momentarily. Thank you for your patience.
These companies can fully utilize our capacity to accelerate their research in vaccine development. Okay, so next slide. So yes.
Please next page.
Okay, next page. So yeah, thank you for your attention and I welcome for any of the questions. That's about it.
Okay, are you done with your presentation?
Yes, I'm already done with my presentation.
Okay, thank you very much. Well, I'm happy to see you back here. I actually got one question. Let me check our live chat. So, we have many viewers. Hello, thank you very much for tuning in. Almost the end of our live talk program. So, Jobs Yoon actually left some message. I wonder what kind of a strategy you had for screening and selecting antibodies, and also if there's any reason to expect the vaccine candidates antigen to have the best defense effect.
Okay. Firstly, for the first question, actually, as I mentioned, with this SVNT kit, it can actually, it is like a mimic of how the virus actually enter into our human cells. So with this kit, definitely it can be used in order to detect the neutralizing antibodies in our human. In fact, the biopharma companies can even use this kit, using this kit in order to detect efficacies of their the neutralizing antibody, as well as to select the possible neutralizing antibody candidates. Yep.
Okay, so you actually answer all two questions.
Can you repeat the second questions again?
Second question is, and also if there is any reason to expect the vaccine candidate antigen to have the best defense effect.
Yes, because there are several different biopharma companies actually develop different kind of the vaccine candidates to come up with the vaccine strategy. So each of the different approach actually consists of the pros and cons. For example, if I'm using the virus base of the vaccine, it probably will it is using the adenovirus for an example. Definitely, it seems to be effective But then the problem is that if particular patient had already infected with the adenovirus before, and then the inside the body actually consists of the immune system that against this adenovirus, then this vaccination approach may probably won't be as effective as the others. So this is very important to select a different kind of the what do you mean, the antigens in order to develop the vaccination.
Okay, thank you very much for your answers as well. So you mentioned COVID-19 in your presentation, right?
Yep.
Yeah, and also we have a special actually session in BioKorea 2020. Have you heard about that? Did you know that? We have a special session with COVID-19 in this exhibition.
No, I actually haven't heard of it.
You definitely visit our website. So
Okay.
we have a special session. Well, for this time, I'm going to promote our peers. Well, we have a special session to share the information and knowledge on trends and current states of technological development to overcome COVID-19. So, yeah, that's why we are holding this event online, and so we have a special session. So,
Can I
please.
ask about is that sessions actually like a discussions with the scientists, the senior scientists in Korea or the biopharma companies?
Not only Korea, but like the world trends and also biopharmaceutical companies relate to COVID-19. So it would be very, useful, very
Okay.
informative. So
Yeah, thanks for your information. I will get my sales managers in Korea to. Okay, yes. Yeah, thank you very much.
thank you very much, Mr. Wang. Have a good day. Thank
You
you.
too. Have a good day.
Okay. All right. So, viewers, we reached the almost end of our program today already. 8th company for today. So it's time to meet our last speaker, Mr. Jordan Blue, Cyclolab. And this company is actively developing several programs in novel applications of cyclodextrin, such as drug delivery system, and their uses as APIs. Besides, it develops innovative and added value formulation with small molecules, custom synthesized and cyclodextrin and manufacture under GMP on the commercial scale. So, hi, Mr. Jordan Blow from Hungary. Hi.
Hi, can you hear me?
Yes, I can hear you. All right. So, how are you doing there?
Yeah, I'm doing great. Thank you.
Close to your microphone because the interpretation service is now being provided. If you speak too close to the microphone, then it causes like white noise. Okay.
Okay.
So, good. Sounds good, right? We're checking sound. Are you okay? Can you just volume down? Can you just volume down? Please?
I'm sorry, can you repeat?
Yes, I can hear you. Can you hear me?
Yes, clearly.
Can you actually turn down your sound? Your voice, your sound is very high, like big.
Okay.
Okay. So say hello to our viewers, Mr. Fluff.
Just a second, I'm not sure how to turn down the sound of the microphone. Is it still too loud?
It's okay, I think. I can hear you clearly.
Okay.
Okay, so say hello to our viewers since you are the last speaker for today and we have many, participants here in this channel. Say hello to, our viewers in your language or English.
Yeah, English probably is better. Hello everyone, my name is Tamas Shohaida and I'm from a company called Cyclolab. We are quite far away from Korea. We are based in Europe, Hungary, and we're dealing with a special kind of molecules that I would like to introduce to you today, and these would be cyclodextrins.
Okay, thank you.
So if we can start, then we can move to the next slide. Okay, so cyclodextrins are technically sugar molecules. They're made of starch, and these are small molecules, oligosaccharides to be more precise, that have a unique shape. As you can see in the picture, they have a shape of a truncated cone or a donut, if you prefer. So they have an inner cavity. And cyclodextrins are hydrophilic molecules, so they're very, very soluble in water. But this cavity that they have has a hydrophobic attitude, and that's why they are able to form non-covalent temporary complexes, inclusion complexes with a different sets of molecules. And this complex forming ability is used in different industries, as it can apparently change the unfavorable physicochemical characteristics of compounds. We can move to the next slide. So this is just a short video of how cyclodextrins compare to different molecules. On the left-hand side, you can see a compound that is solubilized in water, and in the other two vials in the middle and in the right-hand side, it's an aqueous solution of the cyclodextrin. So technically, what you can see and what is the predominant use of cyclodextrins is that they can dramatically increase the solubility of otherwise water-insoluble compounds. This example is curcumin, but they work with a very wide set of molecules. We could go to the next slide. So what happens here? Cyclodextrins, as I mentioned, can form inclusion complexes with different APIs, and this is a temporary interaction, so no new chemical entity is formed, no covalent bond is formed. This is an equilibrium process, so this complex is associated and dissociated on a fairly rapid basis. And what happens is that as long as the active substance is in the cavity of the cyclodextrins itself changes the apparent characteristics of this compound. And this is manifested in a lot of different outcomes. For instance, and the most dramatic is increasing the solubility of the guest compound, but it can also increase physical and chemical stability of the compound. It can mask the taste of the compound. It can improve the permeability of the compound through certain biological barriers. It can thereby improve the bioavailability of the compound. It can reduce the irritation of the compound in certain circumstances, or remove the odor of the compound, and similar characteristics like that. The next slide, please. So in the pharmaceutical industry, cyclodextrins come in different shapes and different types. So we are not just talking about a single compound here. The basic difference is the size of the cavity. So we already addressed that this is a shape of a donut, but the donuts can come in different sizes, and cyclodextrins can come in different sizes depending on how many glucose units compose the cyclodextrin ring. The most typical derivatives are the alpha, beta, and the gamma. Alpha is the smallest, beta is the middle one, and gamma is the biggest one. And these are natural compounds. So these alpha, beta, and gamma cyclodextrin, these are formed in the nature as well, typically upon enzymatic processes by bacteria. This is how they are made in the industry as well. These cyclodextrins are also approved for food uses, these alpha, beta, and the gamma, and pharmaceutical uses, but they have also limitations. because they are not unlimitedly soluble in water, and they are sometimes degraded within the body, and so. So within the industry, we typically deal with the chemically modified versions of these cyclodextrins. In the pharmaceutical industry, there are 7 cyclodextrins that are approved. As I mentioned, the alpha, beta, and the gamma, the native cyclodextrins, as we call them, 3 derivatives of the beta. These are chemical modifications to the beta cavity. One is a hydroxypropyl side chain modified, sulfabutyl side chain modified, and the methyl side chain modified cyclodextrin. And also there is a chemical derivatization of the gamma cyclodextrin, which would be the hydroxypropylation again of the gamma cyclodextrin. So these are the 7 cyclodextrins that are approved in the pharmaceutical industry. Next slide, please. Cyclodextrins are compatible with any kind of dosage form or administration form. These are not new molecules to the pharmaceutical industry. They have been around for at least 2 decades, and there are over 100 different types of products on the market with them. So there are parenterals, eye drops, oral solids, suppositories, topical applications, semi-solids, whatever, any kinds of administration forms. We could say that the typical use in the pharmaceutical industry is solubility improvement, and the majority of the products are parenterals, so typically powders for reconstitution, for injection. The second biggest area would be probably ophthalmics, eye drops typically, and the third one would be oral solids, so in the form of tablets. The mainstream application, about 90% of the uses, I would say, is solubility improvement. The second biggest would be chemical stability improvement, and there are a few other compounds with different applications. We can move to the next one. So what I mentioned so far is the mainstream use of cyclodextrins. Given that cyclodextrin is a small molecule, it has a small cavity, and that small cavity is an ideal fit for another small molecule. So all the commercial formulations that were shown are when cyclodextrins are used to formulate small molecules, and typically that's the mainstream application. However, this is not the only use of cyclodextrins, and the main focus of my talk would not be what cyclodextrins are used for now, but what cyclodextrins will be used in the future. And one of these uses is to formulate biologicals, so not small molecules with cyclodextrins. Given the size differences between the protein and the cyclodextrin, of course, in this case, it's not just one cyclodextrin, but several cyclodextrins, as you can see on the right-hand side, encapsulating certain amino acids of the protein, typically hydrophobic moieties of these amino acids, the aromatic rings. And this is not something entirely new. So products are in different phases of clinics with this, several monoclonal antibodies. And for this particular application, the purpose of using cyclodextrins is not solubility improvement, but to improve the physical stability of the proteins. So they can prevent the aggregation of the proteins, and they can delay the folding. So technically, from a functional perspective, they are an alternative to polysorbates, twins. The main advantage of using them instead of polysorbates is that they have a really excellent safety profile. So cyclodextrins can be dosed in over 10 or 20 grams per day parenterally without any side effects. They are really inert to the body. And they do not metabolize, they do not degrade chemically and physically. They are extremely stable, and they get excreted from the body within 30 minutes, roughly. So that is why, from a regulatory perspective, they're much safer choice to twins, and that would probably give rise to their applications with the proteins. Next slide, please. There are also possibilities to use cyclodextrins in gene delivery, so to formulate mRNA or siRNA, and this application goes over just the simple use of cyclodextrins. It's more like a drug delivery system, because in this case we are not talking anymore about the 7 cyclodextrins that are approved for pharmaceutical purposes, but in this case we're talking about new excipients, and these would be in particularly for gene delivery amphiphilic cyclodextrins. So it's possible to modify cyclodextrins in a way that they contain long side chains as well as positive groups on the other side of the cavity, so they can form micellar like structures, and the cavity of the cyclodextrin would be available to encapsulate certain fragments of an RNA, whereas the group of cyclodextrins as a whole would form a micelle encapsulating the DNA. And this is, again, not a commercial development. It's in clinical phase 1, the most advanced project. But the transfection rate that were achieved with this technology are really impressive, over 4,000-fold improvement compared to common applications. So this can be an alternative to LNPs in the future. We can move to the next slide. Okay, cyclodextrins are also used in certain types of vaccines. And in the vaccine field, there are typically 2 uses of cyclodextrins. One is not necessarily related to vaccines, it's rather related to biotech applications. And in this case, cyclodextrins are used in mammalian cell-based production processes to improve the productivity, the titer, we could say, of the yield of the production. This is because cyclodextrins, certain cyclodextrins at least, can interact with cell membranes and alter the fluidity of these membranes, and that typically results in a much higher productivity, for instance, when one is producing monoclonal antibodies with cells. In the vaccine field, the other application is to use cyclodextrins as adjuvants. Cyclodextrins were shown to be able to boost local immune response when vaccines are given. The most advanced, there is one product on the market from Pfizer that you can see in the picture. It's a veterinary product which uses cyclodextrin adjuvant. And there is another product in clinical phase 2 or 3, which is an influenza vaccine that uses a cyclodextrin as an adjuvant. We can move to the next slide. This slide also relates to, or to better explain you how cyclodextrins work to improve the productivity of certain biotech processes. So cyclodextrins are used widely to make pertussis toxin, which is a toxoid vaccine. And during the making of the toxin itself, cyclodextrins can be used to improve the productivity of the process. And how cyclodextrin does this in this particular case is that when the toxin is created or produced, there are inhibitors in the process that are co-produced. And upon encapsulation of these inhibitors, cyclodextrins can mask the presence of the inhibitors so that the cell would not recognize that the inhibitors are actually in there in the media, and thereby the productivity of the process can be improved. With the use of the cyclodextrin, this particular process, and we are not talking about the development here, this is how pertussis toxin is made on the large scale. is boosted 100-fold, which is a very dramatic improvement compared to the native process. The next slide, please. I've mentioned a lot of potential applications and advantages of cyclodextrins. I should also mention that there are limitations to cyclodextrins when formulating small molecules or big molecules, and that all goes back to the mechanism of action, how cyclodextrins work. And from the very beginning, I've mentioned that the interaction is non-covalent and temporary and then equilibrium-based, which means that if the compound is in solid state, in the tablet or lyophilized powder for reconstitution, the API, the gas molecule, will sit within the cavity of the cyclodextrin. And as long as it's solid, it stays there. Or if you have a solution with a high cyclodextrin concentration, then there will be so many cyclodextrin molecules in the solution that the API will spend most of its time within the cyclodextrin cavity, even if it's in equilibrium. But what happens if we dilute this system, which can happen upon an injection or when swallowing in a tablet that dissolves in the liquid in the stomach? There is a huge dilution immediately, and that drives the equilibrium process towards the dissociation. So the cyclodextrin will release the API that can then bound to proteins or other targets, receptors, or get absorbed or whatever. This would mean that cyclodextrins in themselves are not suitable for extended or controlled release because they will release the API immediately. So when we are trying to achieve a controlled release, we need to combine cyclodextrins with other drug delivery systems, which is luckily a very flexible option because they're compatible with most of them. And these are just a few examples how you can achieve selected or targeted delivery with cyclodextrins. As you can see, you can combine them with liposome, and you can create a liposome loaded with a cyclodextrin loaded with an API, or you can combine them with matrices that degrade slowly. So like this is a hernia mesh on the figure where the degradation of the matrix is the rate limiting step of releasing the API. Cyclodextrins can be chemically modified with targeting agents. So in that case, they can deliver their cargo to a place of action. For instance, in case of anti-cancer agents for tumor cells that are overexpressing certain receptors, cyclodextrins can be conjugated with targeting small molecules that could thereby find these cancer cells. And the last slide is just an electrospun fiber with cyclodextrins, and that's how cyclodextrins could be used for an ODT or a sublingual application. Cyclodextrins are not only used as excipients but also as APIs, and this is the last part of the presentation. There is one cyclodextrin that has been for a long time approved as an API. It's called Shugammadex, and it's technically an antidote which is designed to reverse the effect of certain neuromuscular blocking agents. So technically what happens is that you inject the cyclodextrin and it collects selectively these neuromuscular blocking agents that are administered during surgery. This is a blockbuster drug and the basis of a lot of antidote type of developments with cyclodextrins. Next slide, please. This is just a few ideas of how similar concepts can be used to create antidotes for small molecular heparins, toxins, or different intoxications or different diseases where accumulation of certain compounds is characteristics. We can move to the next slide. There is another application of cyclodextrins to be used as APIs, which is not related to the antidote concept. And for this, the mechanism of action is not really clear. This comes from an occasional or lucky discovery, we could say, where cyclodextrins were used as excipients and they showed effect. So since then, certain cyclodextrins are used in different CNS diseases as an orphan drug. It's called an Niemann-Pick C disease. And it's in clinical phase 3 already, so close to the market. In this case, the mechanism of action is not clearly understood, how cyclodextrins are working or how they are exhibiting their effect. What is for sure that they can treat the disease and they can ameliorate the symptoms, but it's not clear how they are working. We can move to the next slide. So this discovery about the Niemann-Pick and cyclodextrins used not as antidotes but APIs triggered a lot of research, and this is just a glimpse of what is done in this field. Cyclodextrins are evaluated with a wide range of diseases, as you can see: Alzheimer's, Parkinson's, cardiovascular diseases, oncology, and even antivirals. So there are a few papers published recently about their potential use as an anti-coronavirus agent as well. We can move to the next slide. The last slides are about Cyclolab, and I will go through them very briefly. So what we do is we are a small company. We can move to the next slide. We're a small company, and we're typically R&D based. So our internal projects are directed either in reformulating old APIs with cyclodextrins for safer and new administration routes, or creating drug delivery systems for these APIs, or to evaluate cyclodextrins as new chemical entities for some of the diseases or other diseases that were listed on the slide before. We can move on. Next slide, please. So what we do at Cyclolab is that we develop a lot of internal products, and also we manufacture a lot of different cyclodextrins, about 200 different types, very unique ones as well, that can be used in academia or early phase development. We also have a GMP manufacturing plant, or 2 plants to be more precise, where we are manufacturing cyclodextrins for commercial purposes. The next slide, please. So this is just a list of a few APIs, small molecules that are on the market or in late phase clinics with cyclodextrins. And what I would like to highlight from this list is remdesivir. I'm sure that you're all aware of this molecule. This is also formulated with a cyclodextrin, because otherwise it would not be water soluble, and that's how it's formulated by Gilead. Next one, please. And that's just a summary of Dexol, this sulfabutyl cyclodextrin that I already mentioned when I was talking about the different types. So this is the particular compound which is used for remdesivir formulation. Its advantages: this is the safest among all the cyclodextrins and probably one of the most patent, so it can result in a dramatic solubility increase, and a very, very nice safety profile again. So it's a very convenient application for parenteral purposes dominantly. We can move to the next one. Yeah, and that would be it. So I hope that you got a rather clear view about what cyclodextrins are used for now and what they could be used in the near or not too far future, at least. And I hope that it will help you trigger some thoughts about drug discoveries or formulation developments. And if you have any questions, I'm happy to answer now or at a later date. You can see my contacts in this line. Thank you for the attention.
Thank you very much for your presentation, Mr. Jordan Lo, Cyclolab. Yeah, so it's kind of detailed explanation of your company. Though you said it's a small company, but I think it's very high. It has very high potential. So I really enjoyed your presentation. So what are you interested in? What are you expecting in the Korean markets? Do you have any expectation?
Well, the Korean market is rather, so we do not really have too much business with Korea right now. This is a market that we would like to better understand and explore. So we do not really have too many contacts within, but we understand that the pharmaceutical industry is very strong and very innovative, and we would like to primarily better engage with the companies who are in Korea, better understand what they're doing, what is their focus, and establish fruitful long-term relationship with Korean companies. That's our primary goal.
Yeah, through this program, you can definitely have a chance to make Korean company. Thank you very much for your presentation. Thank you.
Bye.
Yeah, have a good day. Thank you. So, viewers, we've got total of 8 companies presentation today. Well, we started this program at 1 p.m. It's been about 3 hours. So thank you very much for staying with us until the end. But it is time to say goodbye. But don't be sad. We have 1 more day tomorrow at the same place at the same time. So our live talk will continue till tomorrow. Don't miss it. I'll see you tomorrow at 1 p.m. Thank you. Bye-bye.